Hello, I'm Professor Fan, and today I'll be live coding a series of spatial transcriptomics data analyses in order to explore why we perform normalization when doing dimensionality reduction and clustering analysis, but not when we do deconvolution analysis using our lab's tool STdeconvolve. So initially, this question was submitted through GitHub issues. And in general, I think it's great to reach out when you have questions, particularly through these public forums where others can see your questions and potentially also see the answers to those questions and even contribute their own answers. So again, this question was about why we perform normalization when doing dimensionality reduction and clustering, but not necessarily when doing deconvolution, at least using our lab's tool STdeconvolve, when it comes to analyzing spatially resolved transcriptomics data. And to really understand why, it of course helps to read through the original paper, to dig into the methods section, perhaps even do the mathematical proof for yourself, which may require reviewing a little bit of your prop stats and stochastic processes. But oftentimes, I actually like to take a more hands-on approach to doing a simulation or an in silico experiment to at least prove to myself that the experimental observations we can make are at least consistent with the underlying mathematical theory. So that's what I'll try to do today. I'll use a simulation to explore the effect of normalization when doing dimensionality reduction and clustering analysis, as well as when doing deconvolution analysis with STdeconvolve. Okay, so why don't I go ahead and share my screen? And yeah, so I think a simulation is particularly nice because we can, of course, create exactly what we need, control for all the covariates and confounders we might be concerned about. And it also just so happens I previously have this simulated spatial transcriptomics data that you can check out on our lab blog. And I'll use the simulation as a starting point and maybe make a few tweaks just so that we can better explore this particular normalization question. So maybe to get us started, we can just copy over the code. And in this particular case, it looks like I am simulating three different cell types, each with 100 cells, and there's 300 genes. I'm really just creating a random Gaussian matrix with an initial mean of 10 and a variance of 10. So yeah, I'm just going to name my rows, gene 1 through M, and the column cell 1 through n times g, since there's n cells in each of the g cell types I'm simulating. And we can just visualize that as a heat map. And in this case, it kind of just looks like random Gaussian noise because that's what we created. But now that we have our three cell types, we can create, let's see, the differentially expressed genes in each that distinguish these cell types. So I'm essentially going to create 100, differentially expressed gene and upregulated by a factor of 100 with a variance of 10 for each cell type, and essentially loop through my genes and add a random Gaussian by the degree of how much I want to upregulate. So this is just going to, let's visualize what that looks like first before running this. Oops. And so this is just going to be the original Gaussian random matrix, but now added with some additional upregulation of genes for each cell type. So red is high expression, blue is low expression, and we can see that each cell type has very distinct, genes being upregulated, just as we simulated. And since generally we have in spatial transcriptomics data, our data tend to be counts, gene transcript counts. So just because we simulated, we simulate things as random Gaussians, there's not necessarily there's not a guarantee that it's positive, and it's definitely not counts because it's Gaussian. So we are just going to make them into a positive expression only by taking anything that's negative, setting it to 0, and then rounding. And this will give us counts, and we can also just visualize that count matrix. And it should, look exactly like what the original simulation was. Okay, great. So this just simulates, single cell gene expression, but now we want to give it a spatial component. In the blog post, I just try to convince myself that these are indeed transcriptually distinct. But now let's make it a spatially resolved transcriptomics data set by creating a set of spatial positions. In this case, a set of 30 by 30 grid spots, and we can visualize this as their centroid positions, and we can just confirm to ourselves, it's a 30 by 30 grid. So this is the underlying spatial positions of our spots, but now we need to essentially put cells in them and put gene expression in these spots. So in the simulation, what I've done is come up with a scenario where I will seed cell type 1 at a particular proportion as we move across the x-axis on this, tissue. Likewise, cell type 2 will have a particular seeding proportion, and cell type 3 will have a different distribution as a function of the x position. So this is just getting all the cells of cell type 1, 2, and 3. I'm going to set the mixture, the percent mixture of each cell type, so that later when so this is just the percent mixture of each cell type as we go across the spots. So this is percent mixture in each spot. We can visualize this as a bar plot if we wanted to. But this is the important part where we can create our underlying kind of ground truth cell type proportions and visualize it. And then, so this is effectively our simulated ground truth cell type proportions in each spot, because in spatial transcriptomics data, at least the multicellular pixel resolution spatial transcriptomics data, we have, each spot may comprise many cells, and therefore, in this case, I've seeded the tissue with only cells of cell type 1, and these spots, mixtures of cell type 1 and 2 in these spots, mixtures of cell type 2 and 3 in these spots, and cell type 3 in these spots. So now, this is just kind of getting the ground truth cell type proportions, but I still have to put, the gene expression associated with these cells in those spots. So I'm going to, make an assumption that there's 10 cells per spot. That seems reasonable. I can even increase this. But now I will loop through the spot positions, sample the underlying ground truth simulated percentage of cell type 1s, 2s, and 3s, and take all the genes expressed by those cells and sum them together, and that will be the simulated spot resolution or multicellular pixel or spot resolution spatial transcriptomics gene expression data set. So what we have at the end, this is our simulated multicellular resolution spatial transcriptomics data. We have the spot mat, which is the gene expression associated with these 300 genes across these 900 spots. And then there's the spot position associated with every spot. Yeah, so this is again my simulated multicellular resolution spatial transcriptomics data set. And you can see that, I have my 3 underlying cell types that I've mixed at different proportions throughout the spots across the tissue. And that is represented in this PCT matrix. But then the actual simulated spatial transcriptomics data set is represented by this gene expression matrix, which tells you the gene expression for this 300 genes across these 900 spots, and then also the spot position matrix, which tells you the positions of each of these spots in X and Y. Okay, so as I mentioned previously, I want to do a couple of tweaks to essentially make normalization or the effect of normalizing or not normalizing more prominent in this simulation, just so that we can really appreciate the effect of normalization versus not normalization. Essentially, I want to make the impact of performing library size normalization so obvious that, that way we can better appreciate its effects. And so in order to do this, I want to essentially change the library size of some of these spots, and then see what happens. So it's as if, let's pretend that I have 30 rows here. Let's say for some reason, every other row, so rows, every other row, I guess starting from the bottom, I will just make their library sizes way, way bigger. So it's as if I sequence these particular rows for some reason way more deeply. And so let's grab these rows. So this will be the Y positions. The Y positions of these spots, if they are in 1, 3, 1, 3, 5, 7, and so forth. So this will give me a vector of Booleans, and then I can figure out which of the positions by name are part of this group. And I'm going to call it tweak. And essentially for these particular tweak spots, I'm going to grab their gene expression, and I essentially want to pretend like, I want to simulate a situation where I somehow sequence these way deeper. So let me create another matrix just so that it's a copy of our original spot matrix, of our original gene expression matrix. But now I'm going to tweak it so that, I want these cells to essentially, or these spots to essentially have a lot more genes. It's as if I sequenced it 5 times more deeply. So I'm just going to, multiply every gene expression by 5 times more. Just to double check I did that right, I can take a column sum, and let's see, we have some spots that have a lot more genes than others. Let me double check that in the original this does not happen. So in the original, they're all about the same, and in the new one, this is 5 times more. So it's as if I sequenced it 5 times more deeply. And maybe just to visualize this, I can yeah, let's see what happens if I just want to double check um, by plotting the original spot positions, and I'm going to color it by the column sums of the spot matrix. And yeah, this is kind of pretty much all the same, not that much variation. You can just double check. There's still going to be some variation, but yeah, it's a very, very narrow range. Whereas if I do the tweaked matrix, now, oh yeah, you can see every every other row is, whoops, every other row is a lot more highly, has a lot more genes. So red is high, gray is low, and we can also just double check. Yeah, the range is much bigger now. Okay, okay, so we have our, yeah, kind of our tweaked simulation. So now, yeah, now it's ready to get to the fun exploration part. Yeah, because what happens if we don't normalize? Okay, we have this data set. I'm not sure if it'll ever happen in real life if this happens, but for some reason, again, some of the rows we have, it's like we sequenced it way more deeply. So now, yeah, what happens if we don't normalize and we try to do dimensionality reduction with PCA and clustering with k-means? Yeah, what happens? So well, let's explore that. So I'm going to just do a principal components analysis on this unnormalized matrix. Let's see if I did that right. Oops, I should have transposed because I want the PCs on the cells, not on the genes. So yeah, let's see. Okay, that's better, or on the spots, not on the genes. So I can, let's see, let's visualize the first two PCs. And yeah, let's just plot the first two PCs. And, oh, well, it looks, well, okay, I'm gonna set the point to, set it to a point instead of a circle. Okay, maybe that doesn't really help too much. But yeah, each point represents a spot, and spots that are closer to each other are transcriptually, more similar. Typically, we would interpret these as, similar cell types. This is kind of a funny pattern, but fine, okay. So now let's do a k-means clustering. I'm going to k-means on the, let's just do k-means clustering. Let's see, how many PCs should we use? Or maybe we should just use the whole, let's see how much variance is explained by the first 100 principal components. Oops, this should be a vector, and okay, well, it looks like most of the variance, I guess, is being explained in the first couple principal components. So because it's a very simple simulation, but yeah, fine. I'll just do my k-means clustering on the first two PCs, and even first three, fine. But I know there's 3 cell types, so I'm going to manually set the center to 3. And let's see, why don't we use a nicer plotting function where I can plot the PCs and then color it by the clusters. Let's see. Okay. Well, yeah, k-means clustering based on this reduced dimensional space. identified, my 3 clusters, which I, in a typical spatial transcriptomics analysis would interpret as my 3 cell types. Yeah, I guess let's see, how do they look in on the tissue? So, yeah, how do they look on the tissue? Okay, so I have my 3 clusters and 3 clusters of spots that, again, I might interpret as 3 different cell types, and they have this very funny, uh, gritty alternating pattern, which actually we know, yeah, this is our underlying kind of simulation. Yeah, this is not particularly close. Yeah, to our simulation. Um, yeah, and actually, well, let's see what happens if we plot this um, again to remind ourselves based on, yeah, plot both of these things, but color it based on the library size. And oh, okay. Well, yeah, so we have a group of cells that have a lot more total genes detected and a lot of spots that have a lot fewer genes detected. And that seems to be driving, yeah, a lot of our k-means clustering. So yeah, so in this case, when we do dimensionality reduction and clustering without normalizing, yeah, this variation in total gene expression and library size affects our downstream clustering and impacts our ability um, to identify, yeah, transcriptionally similar things. So yeah, so let's see, I guess, what happens if what happens if we do normalize? And then in this case, let's see, spot mat, tweak, norm. Yeah, what does it mean to normalize? It means that we are going to yeah, kind of pretend as if we sequenced every spot to the same number of reads rather than having some spots have, 1,000 reads, other spots have 5,000 reads, yada, yada. Okay, so how do we do that? Well, we would need to divide by the total number of reads to convert into a proportion, and then, let's see if I did that right. If I did that right, if I do a call sum, then it should all be 1. Oh, I did not do that right. Okay, so yeah, I must have divided the rows rather than the columns, so I need to do a transpose. Great, okay. Yeah, always good to sanity check yourself. Yeah, now they are 1s, and so this is, you can think of this is the proportion of each gene in each spot. And I guess it's really hard to look at these really small numbers. So sometimes we do a counts per million by multiplying by something like a million to essentially get at how many genes would we have detected per spot if we had a million reads. Okay, so yeah, so now we have this normalized matrix. Yeah, let's see what happens if we just redo all of this dimensionality reduction and clustering, but on the normalized matrix. Okay, so I'm gonna do my PCs again. I want to save both. I'm gonna call it PC2. So I'm gonna do my principal components analysis now on the normalized matrix. Yeah, what happened? What does it look like now? Okay, it still has this funky V shape, but at least now we don't have these 2 groups, I suppose. Okay, so I'm also going to do a k-means clustering again on, yeah, let's stick with the first 3 PCs, fine. Again, I'm going to ask for 3 clusters, and well, let's see what that looks like. Okay, now I have three clusters. And again, I can visualize these three clusters on the reduced dimensional space, but also in the physical tissue space that I simulated. Okay, now at least, yeah, we don't get any more of that, alternating grid type effect. Instead, we get, the clustering analysis identified all of these spots to be presumably one cell type, all of these spots to be presumably another cell type, and all of these spots to be another cell type. And yeah, this is at least a little bit more consistent with our underlying ground truth simulation. Um, yeah, but of course, it's not able to figure out that these spots are actually mixtures of different cell types. Um, at least, at least in the way we've done it to try to resolve three transcriptionally distinct groups. So let's see. Yeah, what happens if we do deconvolution analysis? With something like STdeconvolve. So in order to do that, let's see, I will have to refer to our tutorials. And let's see, so I just want to run the deconvolution on the non-normalized matrix. Let's do a non-normalized first. So let's see. I don't necessarily want to select for certain genes. I want to make sure we compare apples to apples here. So we want to use all the same genes, but I guess now we will just run the underlying latent Dirichlet allocation model to do our deconvolution. I know there's again three cell types, so I'm just going to put that in rather than trying to figure out what's the optimal number of cell types. Again, trying to compare apples to apples. And let's see what happens. And then, of course, I will need to pull out the best model. In this case, I only ran one, so I'll just pull out the k=3 model. And well, let's just copy all of this and see what happens. Okay, in this case, I don't have an annotation. I don't have these particular annotations. So let's see, I will pull out the model and get the deconvolved cell type proportions and their deconvolved gene expression, and I can visualize the deconvolved proportions on my original simulated spots. Let's see what that looks like. Okay, neat. Yeah, so actually, this looks pretty similar to our original underlying simulated cell type proportions, and we're not seeing any funny bands. Even though, again, when we simulated the gene expression, we made certain rows have a lot more genes detected than others. Actually, why don't we compare the underlying cell type proportions that were inferred through the deconvolution to the ground truth, which is this PCT matrix. I guess I could just look at them and see how similar they look. It's a little hard. So maybe I'll just calculate a correlation. And so it looks like the deconvolved cell type 1 has a 0.997 correlation in terms of the proportions across spots to the ground truth simulated cell type 1 proportions. And deconvolved cell type 2 correlates with the ground truth of the cell type proportions of cell type 2 with a correlation coefficient of 0.997. Interesting. And okay, so it seems like even without normalizing, we're doing I would say quite well. Um, Now let's see, what happens, I guess what happens if we do normalize? So again, yeah, there's this normalized matrix. So let's see what happens if we do normalize. And if I do normalize, oh no. Yeah, so one particularity about linear duration allocation modeling is that the underlying process being simulated needs to be it's a count process. So therefore, the entries of our matrix needs to be counts. And in this case, since we normalize to counts per million, these are proportions. They're not necessarily counts anymore. I guess one hacky way could be, well, let's just assume they're counts. We're just going to round it. Well, let's see what happens. Let's see what happens. Um, just for kicks. Okay. I'm going to save the results as LDAs2 now so I can have both results. And again, I'll try to save these as LDAs2, results 2, deconvolution 2, and visualize the the resulting deconvolution 2 when we normalize and hack our way into having a counts matrix instead of proportions. Well, let's see what happens. Okay, so I think that should work. And now, yeah, we have this. Now let's visualize our results. And okay, and yeah, I guess we still get something that looks pretty similar. Yeah, I wonder if we correlate deconvolution 2, how does that correlate with the original? This was the original without normalization. This is with normalization and hacking our way into counts. Okay, well, let's correlate I guess I feel like they look pretty similar. What happens if we just correlate, with normalization versus without normalization? Oh, it's like basically a one-to-one. Um, oh, and yeah, this is in contrast to recall for our dimensionality reduction and clustering. I guess we could also compare, what is this, the clusters we identified without normalization versus the clusters we identified with normalization. Yeah, how do these compare? Yeah, there's definitely a lot of differences. You can see there's no one-to-one correspondence. Whereas, whereas our deconvolve proportions with and without normalization are effectively identical. Yeah, okay, great. So we show that indeed, consistent with the underlying mathematical theory, when it comes to spatial transcriptomics data analysis, for performing dimensionality reduction and clustering, it's really important to do library size normalization, or else the variation in library size could impact our downstream clustering results. However, when it comes to deconvolution analysis with tools like STdeconvolve, whether we normalize or not doesn't necessarily make much of a difference. And again, to really appreciate this, why this is, it is important to understand the underlying math. But I hope this shows that we can also take a hands-on approach in proving these things to ourselves. And in general, I think it's good, as scientists, for us to be skeptical and to always try to find new ways of proving things to ourselves. It's great to be skeptical and to ask questions. It's even better to be able to address your own questions. And I think it's even better if you can find a solution and then teach that solution to somebody else. So with that, thanks for tuning in and have fun tinkering with code.